everybody, and welcome back to another conversation about the science behind the COVID-19 pandemic. Lately, we've been seeing some more numbers coming out from the epidemiological models about what the potential impact of this virus could be. Looking at potentially 100,000, 200,000 deaths here in the US. So today, I wanted to look on the bright side a little bit as we're thinking about what we're facing in the coming months, about 3 technologies that I think are giving us some hope of how we can tackle this pandemic. The first one that I wanted to talk about is one that has really made a splash in the news in the last couple of days, which is a new diagnostic test developed by Abbott Laboratories that could potentially be delivered at the point of care, and patients could be diagnosed and receive a positive diagnosis or a negative diagnosis in as little as 5 minutes, and negative diagnosis, less than 20. This could potentially be a game changer in terms of just speeding up the process by which we're able to know whether somebody has a disease and then making sure we're isolating them quickly. So I wanted to talk a little bit about why this particular technology could be such a big breakthrough and again, some of the science behind it as well. So in a previous conversation, we've talked about the science behind the typical diagnostic test that's out there. And in short, it's basically a test that tries to extract the sample of a virus from a nasal swab or a throat swab from a patient. You then break open the virus, you pull out the genetic material, the RNA, from that virus, and you increase the number of copies of that RNA inside the test tube until you're able to detect whether a certain part of the RNA is present or absent. So this test that's developed by Abbott works basically the same way. It's still taking the RNA out of the virus and amplifying it to increase the number of copies to a detectable level. There are 2 things that are different here. One is that unlike the machines that are used in standard labs, this test is able to amplify that RNA at a single temperature. The normal equipment involves cycling temperatures between high and low temperatures over multiple cycles to be able to generate those additional copies. By using technology that allows you to do that same process but keeping it at a constant temperature, you don't need as much energy to do that process. And so the machine that you can do this with is much, much smaller. It's about the size of a toaster. And the other thing that's a big advantage here is that this machine that allows you to do that is already in widespread use in doctors' offices around the country. It's already used for quick diagnosis of flu viruses and other viruses as well. So it's something that's technology that's already out there, so we can deploy it rapidly, and it simplifies the technology so we're able to get the same level of accuracy in a much, much shorter time. Once the RNA has been amplified, it actually uses a fluorescent signal. So every time that a new copy of that RNA segment is produced, it attaches a fluorescent probe to the copy of the RNA. And so as more and more copies build up, the fluorescent signal grows and the machine is able to detect that. So that's just a quick synopsis of how this technology works. But as these machines are deployed across the country, I think we'll see a real shift in how quickly we're able to diagnose people and then act on that knowledge. So that's really exciting and something that I think will change the dynamics of being able to treat this pandemic moving forward. The second technology that I wanted to talk about is another one that's been making the news lately, and these are smart thermometers. The company that's gotten a lot of attention for producing these thermometers is a company called Kinsa Health. And the way that these thermometers work, they look basically similar to the kind of electronic thermometers that you often see either for home use or in your doctor's office. But what's different is that these Bluetooth-enabled thermometers can then connect with an app on your phone. And under typical use, that app can give you some advice once you take your temperature on, how serious it is, where you can find healthcare. But the added benefit is that when you link up with the app, you're getting location data from your phone that's then synced with that temperature. And so Kinsa realized that they were sitting on this treasure trove of data of being able to track fevers in different locations across the country. And of course, fevers are one of the first and most important symptoms of COVID-19. So they were able to start looking back at their data and start looking at higher incidences of fever than might be expected, and realize that they could actually maybe see these trends in the emergence of COVID-19 before we were getting data from the health organizations and government agencies that are collecting this data on a somewhat lagging timeline. So they've now put their data up online at a website called healthweather.us, so you can see how they're analyzing these data. And what's been encouraging to see over the last few days is that they're starting to see the trends in the frequency of fever start to go down. Now, we are no means out of the woods. We're still looking at expanding testing. Not everybody's using these thermometers, but the data is widespread enough that maybe this is a starting indication that all of the social distancing that we're practicing is starting to have an effect. So in the face of some of these numbers, I think this is some data that is encouraging to people, that hopefully what we're doing is making a difference, and we just need to continue it as we ramp up the testing and the diagnosis and a search for treatment. So that brings me to the third technology that I want to talk about today. So again, in a past conversation, we've talked about the use of existing drugs to try and treat COVID-19, because it just accelerates the speed at which these drugs can be tested and put into practice. But at the same time, another approach to designing drugs is something called rational drug design, where you're actually starting from scratch, looking at the structure of a molecule or two molecules and how they interact, and looking at how you can design a new molecule that could affect that interaction or block a particular molecule. And what's really cool is that this kind of approach is actually being crowdsourced through a computer game. So there's a group that's a collaboration among multiple universities that's developed a game called Fold It. And so you can play this game online. And the goal of this game, actually this game has existed long before coronavirus came along. But the goal of the game is to design proteins or find ways that proteins can fold that result in the most stable structure for that molecule, looking at how much energy is used to put it together. And so when this new challenge came along, the team behind Fold It started creating challenges for any game player to create their own new protein structure and see how well it could potentially fit onto that spike protein that, again, sticks out of the external surface of the virus, to see if we could do this kind of rational drug design approach with the more targets, the better. And so this has been going on for a couple of weeks now in the theoretical space, on the internet challenge. And what's exciting is that within the last week, the first set of crowdsource generated protein designs are now moving forward into lab testing. So through the first 3 rounds of this online competition, people have generated 99 possible structures that are now going to move into laboratory testing to see whether they're potential candidates to disrupt the binding of the virus to the human receptor as a potential treatment. So I think these are just 3 stories about all of the different approaches that people around the world are taking to try and start to attack the long-term game that we're playing in trying to fight the COVID-19 virus. So we hope that to continue telling these stories and bringing these stories to you, but we're going to start also mixing things up a little bit here from the Franklin Institute. Do we have a question? No, okay. So just to wrap things up here, we're going to start bringing you some of our favorite content about stories other than COVID-19, because we know that you're joining us because you're curious about science. So from here on out, I'll be back every Monday at 3 o'clock to give you the latest and answer your questions. But starting tomorrow, we'll be rotating in some of our favorite content from our Spark of Science series and our Mission Material Science series. And my colleagues, Chief Astronomer Derek Pitts and environmental scientist Rachel Valletta and other members of our education team will be rotating in to keep bringing you some of the great content and the great conversations that we're having about all different kinds of science. So please keep joining us. We love talking science with you, and I'll be back on Monday. Thanks.